Hello students, welcome to EPG Pathshala. In this module, we will learn about different biotechnological approaches to biodiversity conservation. In previous modules, we have learned that what is biodiversity, what are its different forms, how does it exist all over the globe, and what are the mega biodiversity country, etc. We have also learned about the applications of biodiversity, as well as we have also learned that what is the need to conserve biodiversity. So, in the biodiversity we have learned that biodiversity conservation is very important. Now, the question arises how it can be conserved. There are a number of ways by which it can be conserved, and one of the way to conserve biodiversity is to use biotechnological tools. That all biotechnological tools we will be studying in this module. So, after studying this module, we will be able to know what are the causes of biodiversity loss. We will understand the need of biodiversity conservation. We will learn about different modes of biodiversity conservation, that is the main theme of this module, and we will also learn about different biotechnological approaches that are available for biodiversity conservation. First, we will try to know about biodiversity conservation. Biodiversity exists in 3 different levels that you have studied earlier also. Just to brief, biodiversity, the 3 levels of biodiversity are genetic, species and ecosystem biodiversity in a given geographical area. Each of the component has its own composition, structure and functions in the environment. In recent past, ever increasing loss of biodiversity has posed a serious threat to the survival of fauna and flora present on this planet. Destruction of the habitat of plants and animals, introduction of exotic species that we will all be learning later on in another module, that is on biological invasion, over exploitation of animals and plants to get the goods, then overpopulation of human and overconsumption of natural resources are the root cause of biodiversity destruction. So, from this all we can conclude that there is a urgent need for the conservation of biodiversity. As far as biotechnology is concerned, this biotechnology offers several new and innovative opportunities for the conservation of biodiversity. Now, this when we talk about biotechnology, this includes tissue culture, micropropagation, marker assisted breeding, conventional breeding, transgenic crops and genomics. These all are quite beneficial for conserving the biodiversity in several ways. Biotechnology has also been used to improve and enhance crop productivity, that we all know that the new varieties have been developed making the use of biotechnology, as well as to conserve and utilize the various aspects of biodiversity. The global concern of biodiversity preservation initiated either by in situ or ex situ methods. In situ methods protect flora, fauna and their natural habitat. On the other hand, ex situ methods involve preservation and maintenance of biodiversity outside their natural habitat. Classical methods of plant propagations have certain limitations in terms of rapid production of plant propagules and their long term conservation. So, the biotechnological methods such as plant cell culture, plant tissue culture, embryo culture, anther culture, etc. are quite applicable and useful techniques for ex situ conservation. Now, in this figure I have shown about different modes of biodiversity conservation. So, one thing we have learned, there may be 2 approaches. One is in situ conservation, that is in the habitat itself, and second one is the ex situ conservation, that is out of habitat. So, in this figure we will see that what are the different modes of biodiversity conservation. And these biosphere reserves may be terrestrial on the land as well as marine. Then we have national parks, wildlife sanctuaries. About national parks and wildlife sanctuaries you will be studying in detail in other module. Then sacred groves. Sacred groves are very old, you can say preservation way of the biodiversity. There is not any module on sacred groves in this paper. I will suggest my students to read about sacred groves, because in recent time lot of attention has been given to the sacred groves as a way of biodiversity conservation. And in ex situ conservation, that is another way to conserve biodiversity out of their habitat, we have botanical gardens, zoological parks, aquariums and home gardens. In India, home gardens of Kerala are very famous. I will suggest the students to learn about the home gardens of Kerala. Then comes biotechnological approaches. These biotechnological approaches are ex situ conservation method, and these biotechnological approaches include gene banks, tissue culture and cryopreservation. So, these are the different methods, different approaches that can be used for the biodiversity conservation. Now, we will be discussing the biotechnological approaches in detail in this module. Nowadays, loss of specific species, that is called as extinction, or a decrease in number of particular organism, that is called as endangerment, are taking place in different of the parts of the world at a rapid rate. Domestic and wild animal species are undergoing a profound erosion process. Conservation of biodiversity through establishment of protected areas like national park, wildlife sanctuary, biosphere reserves, marine reserves, etc., are very effective in controlling the loss of biodiversity. Although biotechnology is commonly thought of as recombinant DNA technology, it is used here in a broader term to include tissue culture, cryopreservation, plant micropropagation and animal regeneration from early embryos. Biotechnology influences germplasm conservation in several ways. It provides alternatives in some cases to conserving whole organism. It can assist with the exchange of germplasm, and the techniques of molecular biology can be applied to the problems of managing and using germplasm, and influence results from the increased demand for germplasm and conservation services by the biotechnologies themselves. In situ conservation focuses on preserving the genetic variation in the location it has been encountered originally, that is in its natural habitats, either in the wild or in traditional farming system. In situ conservation can be carried out at any level in any country without the need for special skills or technology. Properly planned, it can incorporate farmer and industry breeding as well as continued use of resources. Now, ex situ involves conservation outside the native habitat and is generally used to safeguard populations that are in danger of destruction, replenishment or deterioration. Approaches to ex situ conservation include several methods like seed storage, field gene banks and botanical gardens. Ex situ conservation of animal genetic resources includes cryogenic preservation techniques. In cryogenic preservation technique, the collection and freezing in liquid nitrogen of semen, ova or embryos, or the preservation of DNA segments in frozen blood or other tissues. It also encompasses the captive breeding of wild or domesticated species in zoos or other locations away from their natural environment. Ex situ gene banks have similar advantages and disadvantages to those used to hold plant germplasm. In situ conservation enables animal populations to continue to evolve and be selected for use in their natural environments. It is particularly important for species or within geographical regions where cryogenic preservation is not well developed or available. So, the biotechnological approaches of biodiversity conservation come under ex situ mode of conservation. Now, what are different biotechnological approaches which are used in biodiversity conservation? The main objective of germplasm conservation is to maintain constant preservation of germplasm as it can be available at any time. Now, we will discuss various biotechnological approaches which are used in biodiversity conservation. The main objective of germplasm conservation is to maintain constant preservation of germplasm and as it can be available at any time. Biotechnological approaches are imperative for rapid multiplication and conservation of the critical genotype of medicinal plants. These include in vitro propagation through tissue culture, genetic transformation, cryopreservation, DNA banks, etc. Now, let me discuss different tissue culture techniques that can be employed for biodiversity conservation. Different tissue cultural techniques that can be employed for this purpose include plant tissue culture (PTC), micropropagation, somatic embryogenesis and organogenesis, and animal tissue culture. So, we will be learning about these techniques in brief. The details of these techniques you will be learning in your paper of environmental biotechnology. Now, what is plant tissue culture? Plant tissue culture is a quick, independent and efficient in vitro technique to propagate plants under sterile micro environmental conditions. It is very effective method of cloning of plant material and to develop disease free clean plant stock for propagation. Any plant cell has the power of cellular totipotency. Students should know what is totipotency to be differentiated into whole plant in the process of plant tissue culture. The main objective of plant tissue culture is the enhancement of plant production rate by quick regeneration of plants in the absence of seed or otherwise by using the seeds which have very low chances of germination. This figure shows different steps in plant tissue culture. So, here first of all we have a plant and from the plant we take a shoot tip or meristem. Then this shoot tip is grown in the media or in the culture that is called as a primary culture. Students should know that there is not a common media. Different type of media can be used for different plants. Then this primary culture in the second step will lead to shoot development. Actually, auxiliary shoot proliferation will take place. In the third step, rooting of shoots will be there. In the fourth step, we will get rooted plantlets. Then these plantlets from the media are potted, called as potted plantlets in the pot, and finally these potted plantlets develop into full plant. So, these are the total steps. Although this process is not that simple or easy as I have shown here, process is very complicated and very time consuming. Tissue culture techniques can be applied in germplasm conservation of medicinally important plants, important source of pharmaceutical compounds. It can be applied for regenerating different clean disease free stock of plants in the field of agriculture, horticulture, floriculture and pharmaceutical industry to enhance production. Rapid and mass propagation of plant species and their long term germplasm storage can be achieved in a small space within short time period with no damage to the existing population using plant tissue culture methods. Tissue culture techniques are of great interest for collecting, multiplication and storage of plant germplasm and are very useful for conserving plant biodiversity, including genetic resources of recalcitrant seed and vegetatively propagated species, rare and endangered plant species and biotechnology products such as elite genotypes and genetically engineered materials. Tissue culture systems allow propagating plant material with high multiplication rates in an aseptic environment. The second technique that can be used for biotechnological biodiversity conservation is micropropagation. Micropropagation helps in the production of virus free plants and is successful for those plant species which have difficulties in propagation using conventional method. It is an in situ conservation of plant species having special phenotypic characters by introduction of regenerated plantlets directly into its natural habitat. In a short period of time and limited space, it allows the production of numerous plant species. The selection of the explants is very important for the successful micropropagation, which decides the final fate of the developed plantlets via callus or without formation of callus. The micropropagation is rapid and has been adopted for commercialization of important plants such as banana, apple, pears, strawberry, cardamom and many ornamental plants. Here in this slide, different stages of micropropagation are shown. In the first stage, there is introduction and establishment of aseptic culture. Then in second stage, multiplication take place, and in third stage, rooting and preparation is done, and in fourth stage, acclimatation and transplanting take place. So this shows the different stages of micropropagation. Then third method is somatic embryogenesis and organogenesis. The development of somatic embryo from a single or multi cells is known as somatic embryogenesis. Plant propagation through somatic embryogenesis helps to obtain a large number of plants irrespective of seasons and provides several advantages over traditional methods. The main advantage of somatic embryogenesis is the production of numerous plantlets from a single cell, which provides an option for their screening and evaluation. The de novo organ synthesis such as buds, roots and shoots from culture tissue is called as organogenesis. Plant mature cells have the capability to reverse their stage of differentiation and produce new organs under cultured condition. In vitro organogenesis, dedifferentiation and redifferentiation phases are commonly characterized. The balance of exogenous auxin and cytokinin in the medium is essential for de novo organogenesis. Thus, organogenesis entails the regulation of cell division, cell expansion, cell and tissue type differentiation and patterning of the organ as a whole. Now, the fourth technique under plant tissue culture under tissue culture techniques is animal tissue culture. Two principal methods for conserving animal germplasm in vitro are as frozen semen and embryos. Both methods require the maintenance of female animals for either insemination or implantation. Embryos have an advantage over semen because they provide the complete genotype. In some domestic species, embryos for transfer or cryopreservation are collected from animals that have received hormonal treatment to induce an excess of eggs for insemination. The resulting embryos are evaluated for their potential to produce a pregnancy or to withstand freezing. Embryos from cattle, sheep, goats and horses have all been frozen in liquid nitrogen, thawed and successfully implanted. Then cryopreservation is another technique. Basically, cryopreservation is one of the biotechnological methods of ex situ plant conservation and applicable for long term storage of plant genetic material. Cryopreservation is extremely helpful method to conserve rare, endangered and threatened species. There are two types of cryopreservation protocols that basically differ in their physical mechanism. First is classical cryopreservation procedures, in which cooling is preferred in the presence of ice, and in the procedures based on vitrification, in which cooling normally takes place without ice formation. Classical freezing method includes the successive steps such as pre-growth of sample, cryoprotection, slow cooling at 0.5 to somewhere 2 degrees Celsius per minute to a determined pre-freezing temperature, usually in the range of minus 40 degree Celsius, rapid immersion of samples in liquid nitrogen, storage, rapid thawing and recovery. It needs technical skill as it has several steps such as freezing, storage, thawing, reculture of living plant cells during cryopreservation against cryogenic injuries. Maintenance of cryogenic cultures in liquid nitrogen at minus 196 degrees Celsius or in the vapor phase of liquid nitrogen at minus 135 degrees Celsius is in such a way that the viability of stored tissues is retained following rewarming. Various cryogenic methods are being used for various plant species, and these methods include vitrification, encapsulation dehydration and encapsulation vitrification. Basically, vitrification comprises treatment of samples with cryoprotective substances followed by dehydration with highly concentrated plant vitrification solutions, rapid cooling and rewarming and removal of cryoprotectants and recovery. Cryopreservation using the encapsulation dehydration procedure has been very effective for freezing different plant species from temperate and tropical origins. Cryopreservation of seeds is a very valuable strategy for the long term conservation of tropical and subtropical forest species biodiversity, as it avoids problems related to embryo isolation and in vitro handling. In this picture, you will see that there is a tank of cryogenic tank rather of liquid nitration, and how the material is preserved in the cryogenic liquid nitrogen tank. Then another method is DNA banks. DNA banking is also a potential method for the conservation of biological information by preserving the genomic DNA at low temperatures. The DNA isolation is easy and can be used extensively for the characterization and utilization of biodiversity. In gene banks, the plant and animal materials are conserved and are available for breeding, reintroduction, research and other purposes. DNA banks maintain a library of the given DNA sample and provide vital information to the conservation scientists. DNA samples may be of three kinds: total genomic DNA, DNA libraries and individual cloned DNA fragments. The maximum possible genetic diversity of the particular genetic stock can be maintained using in vitro gene bank technique. Some important gene banks all over the world include the Royal Botanical Garden Kew, UK, presently the world's largest DNA bank consisting of over 20,000 DNA species representative of all plant families. The US Missouri Botanical Garden has collection of more than 20,000 plant tissue samples and provide raw material for the extraction of DNA for its subsequent use in conservation methods. Now, second approach is use of molecular marker technologies for biodiversity conservation. Molecular marker technologies are discussed here, include biochemical markers. Different variants of the same enzyme having identical or similar functions are known as isozymes. They are powerful tools for the study of genetic variability within and between populations of plants and animals. They are useful when several taxa, accessions and individuals are to be compared to identify clones and to examine the clonal structure of various plant population. Then we have phytochemical markers. The discovery of phytochemicals from wild plant species is a great achievement toward the eradication of human diseases. With the advancement of modern techniques such as mass spectrometry, NMR, that is nuclear magnetic resonance spectrometry, combined with different separation techniques, facilitated the identification and structural elucidation of these pharmaceuticals. These phytochemical analyses are valuable tools for taxonomic differentiation within species or for evaluating the effect of environmental factors. Variation in biosynthesis of these metabolites could be a result from both genetic and environmental factors, which play important roles in the development of phenotypic variations in plants. Then DNA marker based techniques. DNA based markers are either PCR based or non PCR based. DNA markers assessed genetic diversity based on polymorphism. Potential DNA based markers are single strand conformation polymorphism SSCP, restriction fragment length polymorphism, that is RFLP, random amplified polymorphic DNA, that is RAPD, then amplified fragment length polymorphism, that is AFLP, sequence characterized amplified regions SCAR, then simple sequence repeat SSR, intersimple sequence repeat ISSR, single nucleotide polymorphism, that is SNP. These are either PCR based or non PCR based techniques. So, these all techniques can be used in biodiversity conservation in one or the other way. Then gene transformation. Gene transformation is a powerful tool for enhancing the productivity of novel secondary metabolites of limited yield. Genetic transformation facilitates the growth of medicinal plants with multiple durable resistances to pests and diseases. These genetically transformed cultures can produce secondary metabolites in large amounts and can be promising source for the constant and standardized production of secondary metabolites. Production of potential pharmaceutical compounds using plant transformation strategies and transit expression system such as agroinfiltration and virus infection is known as molecular farming or biofarming. I will suggest my students to learn about molecular farming and biofarming because these are the emerging fields where different companies are earning lot of money. So, students, in this module we have learned that what are the causes of loss of diversity and why it is important to conserve our biodiversity. Then we have learned what is in situ modes of biodiversity conservation and what is ex situ mode of diversity conservation, and we have found that these biotechnological approaches are ex situ methods of diversity conservation. And then we have learned about different biotechnological methods of biodiversity conservation which offer many advantages to conventional methods of biodiversity conservation. Then biotechnology benefits in agricultural productivity and help in the preservation of biodiversity. Biotechnological methods of preservation have potential for long term storage. So, thank you students.